experiences. I worked in AMRI, Curia, and Aragen. And it's been like 6 months. I've joined Advent as a senior application scientist. So today we will be discussing what are all the. Initially, we will be having a presentation of maybe 20-25 minutes, followed by a demonstration. So most of our slides would be on MoleSoft only, the success stories and something. So MoleSoft. So I'll just try to off my video for better bandwidth. Yeah, good morning. And MoleSoft ICM, a comprehensive platform for molecular modeling and drug discovery. So this is something about us. So we Advent was founded in the year 2016. We are a 9-year-old company and we are based out at Pune. And what do we do? We are a prominent distributor of molecular modeling, bioinformatics, and cheminformatics tools. So we provide the first level of support to all the products which we are providing. So very recently we ran a survey with regards to our response. So we were very much appreciated and we were rated 5 out of 5. So So in less than an hour, we could respond to the queries from the clients by 50% and another 35% cumulative by 85 in less than 3 hours. And in less than 24 hours, we could respond to the queries. So definitely not all the queries could be solved in less than 24 hours. But yes, definitely we could acknowledge. If it could be solved, we would take a call and we can directly call to the person and then we can solve the issue. Else, we will set up a Teams meeting or a Zoom or whatever might be. If it is solvable at our end, then we will solve it. Else, if it is not solvable at our end, then we will escalate the same to our US team. So yeah, definitely we will acknowledge that. Yeah, we have received your email and we are working on that. So this is the workflow what we follow at Advent Informatics with regards to the support. So here are our partners. Chemexon, as everyone knows, like Chemexon is a global leader in cheminformatics, chemical structure sketching, editing, and library enumeration, database, structure saving, and many more property predictions. So coming to Compass, which is a tool where you can use your, if you are setting up your experiments, you can use this Compass for setting up a rule-based AI for getting the knowing the synthetic success of that particular reaction. And it also has a module called AutoGrow, where if you have a crystal structure in the pocket of the protein and the system by itself identifies the fragments and grows into the pocket depending on the space. So this is what Compass can be used for. And key module is something where if you have a lot of data in the form of PDFs or other formats where you cannot edit the structure. So this can be used for editing the uneditable structures. SCM is again, we have recently collaborated with which where you can do high-end material science and quantum chemical calculations, quantum mechanics. And Satara for data visualization and Transgene is the only Indian company with which we are collaborated, which provides electronic lab notebook, chemical inventory, and other tools. So MoleSoft, as you see that the complete presentation will be on MoleSoft. And this is something about the MoleSoft. So this is a complete landscape of MoleSoft. As we see, it was founded in the year 1994 by Dr. Ruben Abagyan. At that time, he published a couple of articles on two methodologies like internal coordinate mechanics and bias probability Monte Carlo method. So these two technologies are the base and the backbone of this particular software. So in the same decade, you can see like the early adapters were Bristol Myers Squibb and Novartis. And in the next decade, 2000 to 2009, we also developed multiple tools like Active ICM, Scarab in collaboration with SGC, Structural Genomics Consortium, and a 3D ligand editor which was developed in collaboration with Novartis. And we also took part in so the gray colored ones are the few success stories which we would be discussing in the upcoming slides. So here, GPCR is something where, it was very hard in the early 2000s, there were no crystal structures available. So once the GPCR crystal structure was resolved and the crystallographers threw a challenge to the industry, the one who has the softwares or tools or or a specific methodology and build a homology model of the beta adrenergic receptor, in which MolSoft took part, and we were ranked the top commercial tool for predicting the homology model of GPCR. And we also took part in CAPRI challenge. We were ranked first, and we also developed a specific tool called AP of atomic property fields, kind of pharmacophore, in the year 2008. And in the next decade, 2010 to 19, we also launched multiple tools like MolScreen, which has 2,500 high quality 2D fingerprints and 3D pharmacophoric models. So few of them by default, what we would be getting like ADMET calculations and other things. And we also launched GigaScreen for substructure search and other things. So these gray colored ones we'll be discussing in the upcoming slides. As I told you, in 2020, we also launched RIDE, which is a rapid isosteer discovery engine. This is for ligand-based studies. So we also included a specific scoring function called, now as you know that this being a era of AI, we also integrated an AI-based scoring function called RTCNN to our docking score. And ICM also has been integrated with multiple pharma companies like Arrakis Therapeutics and Elian Therapeutics and many more. So this is the complete landscape about MolSoft. And the global users, you can see one of the you can see here the list of the global users, and this is again the very small subset. We try to put more renowned ones, and there are many more here. Again, you can see Lilly, Merck, Pfizer, AstraZeneca, and again Aravina, Sanofi. So many of these companies are using ACM MolSoft for their molecular modeling and other solutions. So coming to others, like you see in NIH and NCI, even New York University, Stanford, University of Toronto, and USC also are also using the same MolSoft for their research. Coming to the MolSoft users, I believe everyone is aware of the name of this company called Syngene, which is one of the biggest CROs in India. So they use only MolSoft for their complete molecular modeling. I mean, this company completely shifted from Schrodinger to Syngene. And Origin Oncology and Origin Pharma, which are the subsidiaries of Dr. Reddy's, and they also use as MolSoft as one of their molecular modeling tool. Again, Zydus, Think Molecular, and Eurofins, they use their primary discovery tool is MolSoft. Again, we have a very list of very prestigious institutes like NIPER Guwahati, Geetams Hyderabad, and Central University of Kerala, VIT, and many more. So usually the first question that would come in the mind or in the mind of a researcher would be like, I haven't heard anything in the name of this MolSoft. So whether would my work which or would my research which I carry out using MolSoft, would it be publishable? So that's the question what one would arise before buying this or before validating this particular software. So if you go to Google Scholar and type you MolSoft, you can see around 6,000 hits, in which the articles are published into Nature, Cell Death and Disease. Again, Nature, you can see this itself is a full Nature article, which was published by NIH, and it has an impact factor of 69 something, 69 plus. And again, this is a scientific Nature scientific reports, PLOS Biology, International Journal of Biological Molecules, and Cell and Blood. So, and apart from these, there are multiple journals in which the work carried out using MolSoft has been published, like medchem journals like JMC, CMC, CTMC, BMC, BMCL, EJMC, and many more. So what are all the different modules and functionalities we have in MolSoft? So the flagship model of MolSoft, ICM Pro. On top of that, you have some add-ons like VLS and homology modeling. So using these two modules, you can access each and every technique. For example, here you can see there might be an overlap between hit to lead to hit again. So these are all the few techniques which we usually carry out using the hit identification, optimization, lead identification and optimization studies. So these are all the different techniques like flexible docking, 2D, 3D QSAR, pharmacophore modeling, molecular dynamics, R group exploration, virtual screening, docking, induced fit docking, and many more things. So these are all the green colored ones which we can carry out using those two modules. So ICM Pro along with VLS is more than enough to carry out all these techniques. Again, this is the same, just to visualize a different view of the same thing. And what are all the ICM Pro functionalities what we have? So with ICM Pro, you can have ICM Pocket Finder where you can identify the different pockets available in the protein. And you can do regards to docking, you can do small molecule docking, protein docking, induced fit docking, which again has three different types: 4D docking, hybrid partial escape. You can also carry out peptide, RNA, small molecule protein modelings, and MD simulation. And again, you can also generate your 2D ligand interaction diagrams. You can do homology modeling, bioinformatics tools, various tools, and electrostatic. And you can also predict the effect of mutation using Mercy algorithm. And these are all the different things what you can do using ICM Pro. And on top of that, again, database, direct database search. So the interface has been integrated with multiple small molecule and biological databases like PDB. You can access PDB directly from the interface itself, and you can also access UniProt, and you can access NCBI Blast for homology modeling. You can also access Campbell, PubChem, and many more databases. So here, these are the different pocket displays like receptor pocket, ligand pocket, and electrostatic potential. So this is just to show like how good the resolution of the snaps of the graphics that is available with MoleSoft. So coming to ICM VLS functionalities, so almost everything like with regards to VLS, the only difference between Pro and VLS is in Pro, maybe you may have a cutoff of 100 compounds in one go to dock, but here in if you have a VLS, you can dock hundreds and thousands, 100,000 compounds. So like you can do structure-based VLS, fragment-based virtual ligand screening, which has, and again, covalent docking is not included in ICM Pro. Covalent dock with VLS, if you wanted to get access to covalent docking, you may have to have a ICM VLS and again write both GPU and CPU. You can do 2D, 3D QSAR again if you have a VLS more functionality. So this is something about the VLS module. So ICM 3D ligand editor. So this particular tool was developed in collaboration with uh, Novartis, medicinal chemist at Novartis. So what they wanted to do is like exactly usually what happens in discovery, we take out the molecule, we design the compounds on a I mean, we sketch them and we design them and then we prepare them and then we dock them into we generate the grid and dock them. But here it's not like that. So either if you have a complex, the co-crystal along with the ligand or a fragment, or if you dock a molecule and if you can identify like where are the pockets or where you wanted to extend this molecule, you can just edit the structure in the 2D and you can click on this particular button where it will dock and give you the results on the fly. So this can be very much used in optimization studies. And again, it has in the earlier slide, you can see if any of the extensions hits any of the receptor amino acids, then it highlights you that this is a clash what is being observed. And again, it has a very high fragment database, like around 60,000 fragments are there. So depending on the functionality of yours, depending on your requirement where you wanted to control, either you may need a aliphatic chain, small aliphatics, or you need an hydrogen bond donors, or you may have hydrogen bond acceptors. So this way the complete fragment database has been categorized. So you can see this is a small video of it, like you can see how it generates multiple rotamers of that particular aliphatic chain, which is in the solvent exposable area. And again, this way, it is very, very dynamic where you can see what is exactly happening during the designing part. And this can also be used for R group scanning. You can do some isosteer replacements, core replacements, and many more. And again, one good thing here is the complete history, whatever the modifications you do during this particular designing is automatically saved. So you can go back to any step wherever you want it for. So again, on top of that, we have various models like Molescreen, which I told like it has 2,500 high uh, end models. We will be using this. By default package, we'll be getting for ADMET, and this is something called RIDE, which I was speaking about. This is the ligand-based method, which can screen up to 1.5 million conformers per second per GPU on a normal GPU, RTX 4090. So in a minute, it can screen up to 90 million conformers. This is how fast it is. So apart from GPU, on a normal CPU like i7 with 16 GB, 3.93 gigahertz, so it can screen up to 6,000 conformers per second. So in less than 10 minutes, I can screen up to 15 lakhs conformers in less than 10 minutes. And we also have a tool, MolSoft GigaSearch tool, database where you can do substructure search. And again, we also have various tools like for AIML. Rich, this is again, it's a GPU based docking where you can dock up to 100 compounds per second on a GPU RTX 4090. And this is much faster than CPU. And in less than, you can see less than 30 hours, you can screen up to 10 million compounds. And this is one of the fastest structure based docking tool available in the market today. And rich along with the deep learning methods. So we also have something called GigaScreen. And again, we also have Ginger, which is graphical coordinate neural network conformer generator, which is again based on AI. So it generates lightning fast chemicals confirmations. So again, the speed of any software depends on how quicker and how faster the confirmation it generates. So this is how Ginger is making the change or the impact in the speed of this particular software. So these are all a few success stories. So this was published by AstraZeneca, where they compared four different docking tools like Glide, commercial tools, Glide, FlexX, and Gold and ICM. And they said you can see in the extreme right quadrant, like there is a green and cyan color. So the first one being 1 angstroms and the second one being the 2 angstroms. So most of the biological confirmations were less than 2 angstroms. Coming to Glide, they are scattered. They were confirmations beyond 2. And again, you can see RMSD is much more higher in other FlexX and Gold as well. So on a target by target basis, Glide was appeared to be much better for 4 targets, and ICM was much better for 8 different targets. So we also took part in a blind industry D3R docking challenge. So this was started in the year 2016, but we did not take part in 2016. We took part in the year 2017. So this is where they were given a protein FXR ligands, 36 FXR ligands, and they asked to dock these compounds and identify predict the binding confirmation. So MolSoft was ranked first in this particular challenge. We were ranked very first in this challenge. And you can see here in this particular slide. So these are the different people, and this challenge is open for academics, or is open for anyone. I mean, industry also. If they have a specific methodology or if they have a specific tool of their own which they have built. So only thing is they need to predict the biological confirmation of it and submit the data. So here you can see this particular group, they used Autodoc, Vina, Gromax, and they have an in-house ligand interaction energy model. And on top of that, Merck also participated in this where they used Glide, Gold, Amber, and MMGVSC. And again, another group from Merck also used Glide, Gold, Induced Fit. And there is another group from which they also used here. You can see water map, shape screening, DFT calculations, Glide SP, XP, XF model, various models. But still, they could not beat ICM, and we were ranked first in this particular challenge. And this is everything, whatever we are claiming here is available on this website. If you wanted to verify the data, you can always go to this challenge and you can see the data. And in the next year also, in 2018, we also took part in the challenge where it was a different scenario with 6 targets and 5 different kinases. Here also we were ranked first. And in 2019, it's altogether a different picture. Like, there was earlier three challenges were on small molecules, and 2019 it's base macrocyclics. Usually predicting the biological conformation of base macrocyclics is much more tough compared to a small molecule, but still we could predict the biological conformation with a very sub angstrom accuracy. And after 2019 and after COVID, this particular challenges has been stopped. And again, Rampex Pharmaceuticals, they launched a drug called Webomir, which is a beta lactamase inhibitor. So they said, like, whatever the research they carried out for discovery of this particular drug. They used ICM MolSoft covalent docking as their primary tool for this particular study. And again, this is again the the validation of the covalent docking tool, which was carried out using Hungarian, which was done by Hungarian Academy of Sciences. Again, they said ICM was much better with regards to all the available covalent docking tools. Here you can see the success rate of uh, was much more higher with regards to other tools. And ICM also performed with more challenging flexible ligands with 35R heavy atoms also. This is about the benchmarking of the covalent docking tools available from Molsoft. I'll try to skip this slide again. Yeah. So there are multiple success stories associated with the VLS platform. Here again, you can see that the work that was carried out in this has been already published into BMCs. You see ChemBiol, JMC, CMC, JMC, and many more. And here you can see they have screened around 230 compounds, and they could end up with around 456 predicted ligands. And again, 1.2 million compounds were screened, 241 compounds resulting in new class of. So this is just a very small snapshot of only just 10 or 15 articles here. Again, a number of success stories are available on our website, like molsoft.com, vlshtml.html. And this is again a success story. In the year 2013, cloud computing was something. As on today, it's something not so a challenging thing. But in the early 2013, this was something very more advanced technology where Novartis used 30,000 cores on Amazon cloud, and they took up this simulation or computation of millions of chemicals, which on a CPU would might take around 39 years, where they could complete the simulation or the computation in less than 8 hours, and they ended up with 3 Nobel leads. So with regards to technology, so Molsoft keep evolving. And this is again a success story from VLS, where NIH published this article. It was a little older one, but now it's a Synthon based, where the active site is broken down in where a small, I mean, it's a kind of LUDI kind of docking, where you dock fragments into one particular pocket, and then based on the chemistry, based on the pocket size, you extend the molecule into different pockets. That way you fill the pocket. Again, to carry out this complete simulation, ICM is the docking tool which they are using. ICM VLS. This is a very recent article which was published by NIH, and this is again TDP1 inhibitors. So where they NIH used ICM Molsoft's PROTAC modeling platform, and they came up with very good molecules, PROTAC molecules here. So this was the benchmarking which was done by Pfizer and SGC. And these are all the few success stories from our Indian clients where they are using ICM Molsoft. Here you can see Shubhendu Mukherjee and here Arjun, they are using for PROTAC modeling. Here they are using for 4D docking. Here also PROTAC modeling, ADME and PK parameters. And something different what we have seen is Syngene, they are also even for using it for antibody designing also. So this is something different what we have observed. So till now we have spoke a lot about the success stories and many more things about the Molsoft. So what is the exact technology behind Molsoft? So Molsoft ICM stands for internal coordinate mechanics instead of a Cartesian coordinate, and it consists of various features like bond lengths, bond angles, torsion angles, and phase angle. But bond lengths and planar angles are considered to be rigid here. Only torsion angles are given for degree of freedom. That's the reason this particular software is much faster than other tools. And again, on top of that, for the simulation, it also used bias probability Monte Carlo simulations to globally optimize the ligands in the grid potential maps of the protein pocket. And these are all the 3 different scoring functions what we have. Unlike other softwares, we have only one scoring function. We have ICM score, which is a physics-based scoring function. We have a statistical scoring function called MF score and RTCNN. So we'll be discussing more on this RTCNN, which is an AI-based scoring function. And this scoring function was trained using a putative ligand complexes that were taken from PDB, so that. And here, one key thing about this RTCNN is it doesn't give more penalty to the induced fit receptor clashes. So how it would be useful is when you are doing a VLS screening, for example. So in that case, if you want picking the molecule or prioritizing the molecules based on one score is not suggestible. Here, you can plot the data with regards to RTCNN as well as the normal physics based one. And then we can take the subset of them which are better ones. So we will see this particular plotting technique during our demo. And it has RTCNN has outperformed with regards to various scoring functions. Here you can see Autodoc, Vina, Glide, and even Discovery Studio, PLP, and even other tools also. With regards to the predicting the top 1, 2, and top 3 biological top 3 confirmations. And we also have a technology called APF, which I was telling it was developed in the year 2008, where it has various features like hydrogen bond donors, acceptors, sp2 hybridization, lipophilic small, and again, this is a electrostatic, electronegative and electropositive characters. And in induced fit, it depends on situation, which kind of flexible docking which you would perform. Herein, we have three different methods like 4D docking, hybrid, partially explicit, and scare. So with regards to 4D docking, if at all, for example, if your active site is much flexible, for example, there is a lot of conformational changes when a ligand bind, when an agonist bind, when an inverse agonist binds. So in that case, for the dynamic conformational changes, if you wanted to distinguish your molecules from an agonist to an inverse agonist, so you can always use the information. If you have the crystal information, then you can merge those and create an ensemble of those crystal structures, and you can carry out the docking of your ligands for distinguishing the agonist versus inverse agonist. So this can be used for 4D docking, and it has various other applications also. Hybrid partially explicit. This is something where you give a degree of freedom to 3 to 4 amino acids. For example, usually when we are carrying out our research, suppose we might find a kinase with a DFG in with a ATP. So there might be a ligand or there might be an active compound which is type 2 inhibitor or DFG which binds in a DFG out conformation. So the crystal, that particular crystal structure is not available in the PDB. So usually what is done, either we take that structure and build a homology model of DFG out conformation. But here we can select those three amino acids and dock the molecule where it will generate multiple conformations of that particular loop. So in that scenario, we can use it hybrid partially explicit. And scare method here where it is scanning of alanine and refinement. So where here it uh, two alanines at a particular point, it replaces them, it replaces the original amino acids with the alanine and it docks your compound, and again those alanines are replaced with your original molecule, original amino acid. So this way it will try to generate the conformations of your receptor. Coming to other technologies, what we have optimal docking areas for protein-protein docking. So what it does is it tries to identify what are all the instead docking your, this is a protein-protein docking where instead docking on your protein throughout the complete protein interface, it will, this particular algorithm will identify which could be the most possible area. So this algorithm will identify that, and it will, based on that particular optimal docking areas, we can dock the other protein into this particular area you can see here. So the red colored ones are the optimal docking areas. And here in this, it uses fast Fourier transform algorithm, which is a very simplified. Initially, it applies a simplified scoring function followed by a very complicated scoring functions like electrostatics and other things. So you can also calculate the predict the effect of mutation using a Melzi algorithm. And here you can find out the using ICM pocket finder where you can find out various receptor pockets that are available in the protein. You can also predict the reactive cysteine residues and covalent docking. So this is something what I have for the different technologies what we have. And this is how the interface it looks. And this is the interface. This is what we call it as a header and workspace and a 3D graphical window and terminal window. So if you are an advanced user, you can write your code here and you can execute the same thing when which you can. You are not, if you are an advanced user. So this is a selection button. So this is something about the presentation. So if you have any questions after this, I would be very much happy to answer them. So after this, we will quickly jump on to a demonstration.
Thank you so much, sir. It was great to
Yes.
learn all those advancement and how ICM Pro actually revolutionized our this entire drug discovery process. And it's so
Yes.
great to see all those coding function benchmark how the software showing much more better activity and much more better performance compared to other all the paid and open source software. So it is so great. Congratulations, and I hope our students got some idea, and our faculty
Yes.
members also got some inspiration from this. So I have one question, sir, only regarding the
Yes,
4D
please.
docking you have mentioned.
Yes.
So
Yes.
in 4D docking, whether if a protein got mutated, so whether we can also take that one, or we may take all the commonly available, because the problem is happening when
Mhm.
we take some predicted structures like AlphaFold or like Bolt2 and some experimentally taken structure. The structures are so much different due to their less energy minimization and things. So what will be your
Yes.
comment in that regard?
Sir, here it can be used in multiple ways. For example, now if you have a crystal structure of a wild type, right?
Right, right.
Usually, yeah, wild type, and you have multiple crystal structures of different mutants. Now, you can make an ensemble of all those crystal structures into one crystal as a one protein, and you can generate the grid of them. And you can
Right.
dock your molecules, and you can see. So, for example, if you have three or four different mutations with regards to the wild type, and when you generate the grid for all these four proteins, instead docking individually, in one shot, you can dock all your ligands, and it will tell you this particular ligand is docking onto which protein, receptor 1, receptor 2. It will rank them based on the receptor. So, this molecule binds very well into your mutant 1. This molecule binds very well into your mutant 2. And not only for mutation, even if you don't have a mutation, even in wild type, for example, you have multiple crystal ligands also. In one case, you have a small different chemotype, the other one has a different chemotype, and you see a lot of side chain rotations. When some big ligand binds, or I mean, the side chain conformational changes would also be there. So you can create an ensemble and you can address those issues also.
Great, sir. Great.
Yeah,
Thank you so much, sir. So we
yeah.
can proceed for the demonstration purpose, and if anything more you want to share.
Yes, thank you.
Welcome, sir.
So I believe everyone is able to see my screen.
Yes, sir. Your screen is visible. Please
Yes.
go ahead.
So this is how the visualizer look. I mean, this is in a different color, which you can make it based on your interest. So this is a default visualization one. And if you are working at night, you can make it to something else. This is just a small thing which I thought of showing you. And in the presentation, I was telling like it has been integrated with multiple databases, right? So this is PDB integrated with PDB, NCBI Blast, UniProt, and various other PDBs, Sequence Search, Homologs, you can search, PubChem and ChemWell, Drug Central, and it also been integrated with these various things. So, like for example, let's see, now you started your research on a specific cancer and you are trying to identify the different proteins that are involved into that area. For example, you are looking into liver cancer, you are trying to target kidney cancer, or you might be working on a breast cancer. So let's assume you are trying to work on breast cancer, you are trying to identify which is that particular protein if I would target would be a very better protein of interest. So in that case, for example, I did my homework and I identified like PARP1 could be the very better protein. So PARP1 stands for poly-ADP ribose polymerase, which is a DNA repair protein. So just typing the correct name of this protein and entering this button, either enter or you can hit this button. So it will get you all the crystal structures, the all the information that has been deposited in the PDB. So you can see you can sort them based on the ID, just like in Excel. You can also sort it based on your experimental methodology. And you can see here, other than X-ray, you can also see NMR, electron microscopy, and even if it is cryo-EM structures also, it will get you all the information. So again, you can sort it based on resolution. Usually what we do during research, we try to take the lowest resolution structure, and you can also sort it based on date and multiple things. For example, here in this case, even it will also give you the information of small molecules also, with which it has been co-crystallized. So if at all I wanted to move this molecule to after experiment, and I want to work on a particular chemotype, then I can either pick this crystal structure, or I can, depending on my core, I can take the right uh, crystal structure for the study depending on my new designs. So this way, if at all I wanted to see, as I mean, I know the PDB-ID, then I can just definitely type the 4-letter code of it. For example, 4R6E from here, and I can download it. Else, if I wanted to search it from here, I can type Control F. I can say, just like in Excel, it will take me to this. And if I wanted to load this, it will load this particular protein in my terminal window, graphical window. Yeah, this way. So this is the way we can just have a look at all the crystal structures that are there. Again, this selection by itself, going to PDB and searching all the different PDBs with regards to various parameters, again, it's very cumbersome task. But here you can just do, you can download everything just in fraction of seconds. So the other question is, can I search it with only the PDB IDs or the protein name? Definitely not. There is a structure sketching tool here available, right? So usually medicinal chemists or synthetic chemists, usually they try to work on a specific core, like, okay, I want to carry out my research on quinazoline ring, this particular ring, and I want to search what are all the different crystal structures that are already reported, substructures or substructure search. Here it's a substructure search. And I want to download and see what are all the different proteins or different diseases it is targeting. I can just send it to Smiles and hit this button. And then this way, it gives me the information of all the structures from the PDB, like around 70. This is 70. No, this should be a bigger number actually. So this is how it helps us in identifying what are the different cores that are on which I can start working. For example, now let's see this particular core. It is available in the particular target called checkpoint kinase, and this is again with functional role on of HSP70, and this is the same core is having some use in some importance in Trypanosoma brucei and Escherichia coli. So here, for example, if you wanted to target couple of, I mean, now with this, we will know have some information like, okay, if I have this kind of core, I can try targeting 3 different proteins coming from 3 different organisms. One is from humans, Trypanosoma and Escherichia coli. And whatever my new theoretical designs which I would be making, I can dock into these targets, 3 different targets, and I can see which of those are showing good docking scores and good interactions. I can select, I can prioritize my molecules based on different ways. So this is one way we can use this information. So what I'll be doing is here, we will prepare this protein and we will redock this particular crystal ligand into the same protein and see how, what would be the RMSD and how speed and how efficient it is. So as you see here in the workspace, there are multiple chains associated with this protein. All those chains are, it's a homomultimer, 4 different chains, all of them has the same crystal ligand. For that reason, I'll be deleting everything, keeping one chain, I'll delete all the other chains. Now, for example, I want to see the ligand, bring it to the center of the screen. What I need to do is double click on this ligand. And again, if I want to see the structure, I can just click on this. I can see the 2D structure. If I wanted to see the sequence, I can just click on this and see the sequence. So I'll double click on my ligand, which you can see is selected in the green color, and hit this zoom button. Now you can see this is the crystal ligand with which this particular protein has been co-crystallized. Now, again, as you know, this is a XR structure clearly seen here. You don't have hydrogens. So in like all other softwares, we may have to prepare this protein and convert this protein into an ICM format. So before that, you can also see something adjacent to this ID, PubMed reference. So it is just you can click on this button and it will take you to this particular article. And if you wanted to understand more and read more about that particular crystal structure, you can always download this article and read more from here. And with regards to UniProt also, you can just click on this. So this is very highly integrated with all these tool things. So now let me prepare this protein and convert this protein to an ICM format. For that, I'll give a just right click. So I'll say convert PDB. Anyway, I have deleted all the different water molecules. If there are any water molecules which have an importance, like if any water molecule is in the pocket and making 2 to 3 hydrogen bonds, so we may have to do that analysis before we start the docking. And here we can see various options like optimize hydrogens. So during this protein preparation, it will add the hydrogens and then it will optimize those hydrogens for better hydrogen bonding network. Coming to optimize histidine, proline, aspargine, glutamine, and cysteine. Histidine usually exists in 4 different protonation states, couple of them in acidic and couple of them in neutral. So it will try checking all those protonation states and keep the best ones. And again, aspargine and glutamine, it has a amide in their side chain. It will try to flip the side chain, the amide 180 degrees and see how better hydrogen bonding network is. And again, cysteine, it forms a covalent bond. Either it can be a protein, two cysteines in the protein, disulfide bond. To recognize that, it will also optimize that. Or else some of the cysteines might form a coordinate covalent bond with your metal. In that case also, this will be useful. Again, there is no science with regards to these two options. Replace original, display the result. Replace original is X-ray structure will be replaced with ICM. If we uncheck this, then you will see an X-ray structure and ICM structure, prepaid structure as well. To avoid confusion, we will check this. So this will display the result here. Again, hide missing side chains is something during crystallization, crystallographers may not be able to understand particular side chains of few of those amino acids. So if we uncheck it, system will build the side chain and it will optimize that particular side chain. But I believe this being 2.2 angstroms, I believe almost all the side chains might have been nicely derived. So assign secondary structure. So we are telling the information to the systems key. There is a helix, right-handed helix here. There is a beta sheet here, and there are loops here. So that information it captures and predict formal charges. ICM Allsoft has an inbuilt pK model where it predicts the formal charges of the ligand at a neutral pH and optimized ligand tautomer. So here there is no chance for ketoenol tautomerism, but yes, definitely if your ligand has a ketoenol tautomerism, it will try to generate that also if you check this option. And then we will hit OK. So this is just one single thing, like give a right click and convert. It hardly took 10 seconds maybe for me. Now my protein is prepared, right? So now mostly here you can see prepared means the hydrogens are added at the you can see on the nitrogen. And now I wanted to understand what are all the different interactions this particular ligand is making. To analyze that, I can give a right click, go to pocket, and I can click ligand pocket interactions. And now you can see here, this particular molecule is making hydrogen bonds with glycine here, one with serine and the other side with aspartic acid. And unlike other softwares, here you can see the hydrogen bonds in different colors. In other tools, it is only one way of representation, green color. So here in ICM, red colored hydrogen bonds are considered to be the strongest hydrogen bonds, and followed by green color, which are moderately energetic, and blue color are less energetic. Means, for example, let's consider the red colored hydrogen bond energies range in the range of -7 to -10 kilocalories and -5 to -7 would be green. So that way it has been categorized. And again, one more question would come into the mind. Here you can see, even in blue, you can see two different sizes of the spheres, right? In this particular category, the bigger the sphere size, so this is much stronger than this particular hydrogen bond. So this way we can understand just by visually by looking at the hydrogen bonds by itself. So does a ligand make only hydrogen bonds in a protein? Definitely not. It will also make certain hydrophobic or pi-pi stacking. So if I wanted to understand what are all the different pi stacking interactions, what all I need to do is click on this button, display pi-pi stacking and pi cation. And now here you can see this benzopyrazole ring has been sandwiched between these two aromatic amino acids, pi-pi stacking. And on the extreme right, this piperidine ring is also making pi cation interaction with this aromatic amino acid tyrosine. Now this is something usually we carry out do this kind of interaction profile before you do a docking of your own molecules. Now on top of that, if I wanted to see the active site, what I can do is I can give a right click, pocket, receptor pocket, and I can click OK. So this way I can generate the surface of the pocket just by single click. I can change lots of things. So this way for uh, snaps after my docking, I can take this kind of good resolution snaps. I can keep the fogging on and fogging off. I can put some stereo and I can make multiple things, HD, I mean shadow kind of thing. If I off click on this shadow, so you will see this kind of shadowing also. So just by one single click, you can change your graphical visualization like whatever the way you wish for. So this is just something about visualization. Now let us quickly uh, dock this molecule, prepare the grid. Now we have prepared this protein. Now we will be preparing the grid. For that, what I would have to do is I will take this molecule out of this pocket. I'll give a right click. I'll say move from object till now, and I'll try to rename this with crystal ligand. Right. Now, before docking, we have to generate the grid for that. For that, what we have to do again and again before docking, I would also like to show you how to run a ICM pocket finder. Consider now this is an apoprotein. Let's consider that this protein we don't know where the active site pocket is. So if I wanted, or this could be an homology model, or this could be an alpha fold structure. So whichever it might be. So if I go to tools, 3D Predict, ICM Pocket Finder. So there is something called tolerance 4.6. So this will tell us identify the deeper pockets. If you are working on a shallow pockets on the surface of the protein, you can decrease this tolerance to 2.5 to 3. And but for this particular thing, we'll keep it default options and we'll keep there are multiple other things like display results and keep compounds. So if at all there is a crystal structure with the ligand, if I check this, then what it will do, it will not read this pocket and it will try to identify other pockets like this being orthosteric pocket, it will try to identify in other allosteric pockets that are available. So we will keep default things and say okay. So this is something called sitemap. And again, you can see here, this has identified six different pockets and they are ranked based on specific things like the volume. You can see the hydrophobicity, the buriedness, aromaticity of this particular uh, pocket and certain B factor and various other relative factors. So based on these various uh, parameters, it has been ranked number one based on delete score. So this delete score was developed in collaboration with Merck, where let's see, let us, I will try to check these different active site or the pockets cavities which it identified during this simulation. So these are all the six different pockets it identified and it ranked based on the size and the drug likeness score. So I'll try to off the other pockets and again the representation also I can change. So I can what I just have to do is give a right click and change it to dot format this way. So let me put my crystal ligand which pocket it is capturing. So this is the pocket which it correctly identified the pocket and it ranked this particular pocket as the first one, first rank. So other than dot, I can also convert it to wire format. So whatever the different various visualization ways I want, I can do it. I can keep it to smooth, transparent as well. So these are all the different ways of identifying the pocket. Now we will quickly generate the grid. We'll double click this. We'll go to docking, new project. I'll try to keep things default. This is the name of the job. So this should not start with character numbers and it should have minimum 4 characters. And this is the path where you wanted to save your job, right? And this is the receptor protein. Usually now we see we have only one protein in the workspace. If you have multiple proteins, so you may have to select the right one using this drop down. Now again, you have defined site around the selected ligand molecules. Now, the reason for selecting this ligand is we are trying to assign the pocket of residues which are in the active site pocket. Now we have selected the different active site residues and we are click these receptor maps immediately and we'll say OK. Now you see it has generated, it has kept a probe in the center of it. This is called a probe. And in others, we consider it as a centroid. So this can be used for, it is suggested not to move, but if you are doing some fragment based docking studies and now you wanted, suppose this pocket is much bigger and you wanted to dock your fragments into this area only. In that scenario, you can move this probe into the other side of the pocket and generate the grid. But here we will keep it by default where it came and then we will hit on go. And like other softwares, the external grid is in cyan color. What I can increase or decrease the size just by holding any corner from my mouse. I can increase the size of the grid and I can decrease it. But with other tools, you will get XYZ coordinates. So you may have to adjust them. Here it is like very freely you can move any of those things. So now when I hit go, so now this is generating the grid maps in the backend. So yes, it finished. The grid maps are generated. How would I know? Because the name of my job along with underscore REC. So when you find this particular thing in your workspace, so the grid maps are generated and they are ready to do the docking. Now docking a molecule which has 3D coordinates doesn't make sense. For that, what we will do is we will extract this ligand. We will give a right click, extract the ligand. This way also we can extract that into a 2D. There is another option. For example, we'll go to this 2D sketcher and I know that this molecule, whatever that is being co-crystallized is a drug molecule called niraparib. So if you know the name of the drug, you can type it here, search NIRA. So this is the structure of that particular drug. As I told that this tool is integrated with Drug Central also. So this will give you quickly the structures. And again, in the right side, you can see in this box. So it has calculated the various parameters of this particular molecule like formula, SMILES, IUPAC name, INCHI, acceptor, donor, and any alerts are there. It will mention you PSA, TPSA, and various other things. Right. So anyway, we have extracted this ligand and then we will quickly go to docking and launch the dock chemical. So I will off this and we will go to, this is the table which we wanted to dock now. We'll say okay. There are few things which I thought of we will be discussing here. Thoroughness is something how bigger you wanted to run this simulation. In this case, we will keep it 3 small molecule, 1 molecule docking. When we do VLS, we will keep this thoroughness as 1. Means virtual ligand screening, which is fast docking or rough docking. So if you wanted to do more thorough docking, we may have to increase this thoroughness to 5. Or as I was discussing, when you have 4 different mutated structures and one wild type embedded into one particular protein. So we may have to increase this thoroughness to 10. Why? Because it will try all the the simulation, it will try in all the protein pockets, then it will give you the output. And again, if your molecule is much flexible, much more flexible, you can increase this thoroughness. So these are all the various situations where we can increase the thoroughness. And number of confirmations, I will keep it default. So sample double bonds, it will try to, if resonance, if it is there, it will adjust them. Sample racemic. And you can see this structure, 2D structure in this 2D window. This is one type of isomer. This, I mean, it is a crystal structure, which is one type of isomer. For example, if this is a racemic structure, so what I can do, I can check this. So this will automatically generate both R and S isomers. Or if it has multiple stereo centers, it will generate multiple trans and cis and trans isomers, and then it will dog them. But I will not check this because this being one type of it. So sample tautomers, as I told that it will also generate the tautomers. So CNL protocol is already embedded in this. So this is just something more about this particular protocol. We'll hit dock, and now the docking has been started. And to see where the job is running, go to Windows, background jobs, and it has launched the docking. So here one thing is there. Yeah, it took hardly 16 seconds to finish the docking, and I'll have to say okay. It will load this particular chem underscore docked. And again, what I was saying, I couldn't check that. Yeah. So now this is the docking output. Now let us analyze the docking output, how it looks. So in this table, if I click anywhere in this particular table, so this is my docked confirmation. But the graphical viewer is graphically, it's not looking good. So with few clicks, we will convert this to a very good way. To do that, we will click on this button, uh, extra panel, and we'll go to tools. And then we can also go to visualizing. We keep the pretty surface and say visualize. So this way it changes the visualization in one way. And still I cannot see the protein and multiple things. For that, I'll go to lightning, decrease the transparency and decrease the brightness. So now this way I can see the protein. So again, I can convert it into multiple ways. So I'll off this way and I can change the protein representation into different ways by clicking on this button. I can change it to ribbon and I can change it to smooth ribbon, and even I can change it to secondary structure as well by secondary structure this way. And I can off this. If this is the transparency, I can off it and on it. So this was on. So that's why it was looking like that this way, transparency. And again, one thing you can see the hydrogen bond, even you can see the hydrogen bond distances are there here. Now we have finished the docking of our crystal, redocking of the crystal ligand. Now I would like to overlay the crystal ligand with the docked conformation. So the cyan color, blue color one is the crystal ligand, and the yellow color one is my dock ligand. So visually looking, this is pretty very nicely overlaying. But to quantify that, I want to calculate the RMSD. To do that, I will go to tools, analysis, RMSD. Here I will go to chemical and I will select the ligand conformation. This is a crystal ligand versus my dock conformation. I'll say okay. So the RMSD is close to 0.34, which tells it is very good. Anything less than 1 is considered to be better. So this way we can dock it and we can generate our confirmations. So you can also change the residue representation from single letter through 3 letters just by clicking one single button. And you can also drag the labels. I mean, drag the labels means you can just move it based on your for your snaps, and you can move these labeling from to C alpha. You can move the labeling to side chains this way. I mean, various way you can play around here using just with your mouse. So this is what we have finished with the redocking of it. So do you have any questions here, Doctor? Uh, uh, Sorry.
No, sir, please go ahead.
Yeah. So now what we will do is, now anyway we finished the redocking. I got a very good redocking of my crystal ligand. Now I want to download some dock my designs, or I want to download some molecules which are already published on PRP. So if I want to download them, what I can do is I can quickly go to mole screen, find Campbell PKD from mammalian targets. So this data table, this is for only mammalian targets, not different organisms. If I'll click on UniProt and type the name of the protein PARP1, enter. So here in this table, you can see, I'll check this table. You can see here, number of compounds is around 5,500. So here these structures are already biologically tested and the data is available on Campbell site. So to download these structures, what I need to do is only just check this and get PKD data from for the checked targets. So this way I can just in a fraction of seconds or minute, now it has downloaded all the data with regards to PARP from Campbell. So this is how fast you can gather the information from Campbell also. Now, if I don't have a specific field called molecular weight to sort these compounds, I'll go to chemistry, calculate properties. Here there are various properties of your interest. Like you wanted to calculate pK, mole, I mean log P, molecules log P, IUPAC name, molecular weight. You can check anything whichever you wanted, and we can just say okay. So now it has calculated. To sort it, I'll just hit on this button. So this way we can download the data, and again I'll do is I'll query something from here from this complete struct database. I wanted to do a substructure search and get all the molecules which are having a substructure of my drug. So what I can do is I can quickly sketch the structure of that molecule substructure this way, and I'll select this as a Campbell target, and then I will say search. Right. Now you could see I did a substructure and I got around 80 compounds from here. So this is how easily you can download your data. For example, now if I wanted to dock these molecules as an SD file for VLS, what I can do is I can quickly give a right click, save selection as, I'll select desktop, I'll save this as PARP. Uh, VLS. I'll name this as PRP VLS. So now this SD file is ready for me to dock. And now again, in docking, we'll go to batch ligand from SD file, go to browse wherever we have kept this SD file. For example, PRP VLS, say OK. Okay, now here you can see docking effort as 1. So as I was telling during docking, this effort was, tolerance was 3. Here by default for VLS, it is 1. And again, we can use some filters. For example, now here in this case, consider the docking score of this particular redocking of this ligand was minus 46. Now I want to see the ligands which have the docking of greater than minus 40 only. So I can select this option. I can put this 40 and I can say, I can increase the number of parallel jobs. I can say, okay. So this way I can launch a VLS job. So now we will be this VLS option is available in almost every single other tool, but there is something specific with this software which we can do. So I will try to show you that how we can do that. For that, what I will do is, we will generate a pharmacophore and then using this particular core, and we will screen this database with the pharmacophore and do the docking on the fly, everything. We'll give a right click. We will go to edit molecule, and then We will save this. It crashed. Sorry, it crashed, but anyway, I can prepare it quickly once again.
ये सब दिल्ली माना जाता है ये 8
I'm trying to redo the same thing what we did.
If any of the participants have any questions, they can ask or type in the chat box.
Yes. So, what I thought of showing here is, so apart from normal docking, we also have multiple other options where we can give this particular. Now we will generate this as a template. For example, we'll go to docking and we will assign this as a template for docking. So this is our template called a.form and we will give a pharmacophore APF and then we will say okay. Now we'll go to chemistry APF consists pharmacophore. We'll generate this one. So now we generated a pharmacophore of this core, small part of this core. Now you can see all the ligands are in this way they are extended. So now when I crop this molecule with a particular core, I'm also seeing an additional pharmacophoric feature here. But if I put this, when I screen the ligands which I retrieved from that SD file, so they will not match. For that, what I will do is I will crop this. For example, here you can see different features like hydrogen bond donors, acceptors, positive, electronegative, nothing is there, aromatic. Now, if at all I want to, these two acceptors I want, but I don't want this donor feature. For that, what I can do, I can give a right click. I can split it to two. The first donor I will keep, the second donor I will delete. I'll give a right click, delete. I'll delete this donor also. Now, my pharmacophore which I generated will have these features. Right? Now, what I will be doing is we will dock the new molecules, whatever we have downloaded them for VLS. We will screen them first with pharmacophore and then dock them automatically. So this is one time it will do that thing in the one go only. So docking effort, I'll keep 1. I'll keep the value as minus 40. Number of parallel jobs 3. And then I'll say okay. So now in normal VLS, it is docking only into the active site. Now what we are doing, we generated a pharmacophore in the protein active site. First, those molecules are screened with these pharmacophoric features. Whichever that are passing with these pharmacophoric features, are then docked and it will give the output. So we will see this, what is the scoring in this. And again, now you can see in VLS, this is a small set, 50 compounds, 80 compounds. It will happen very quickly. But in case now you are docking 1 lakh compounds or 10 lakh compounds, usually in other tools, what we will do, we will launch the job. It might take 10 days. Then we will have to wait for 10 days and come and see the data. But here, it's not like that. What you can do is we can go to quickly docking and we can make our own hit list. So whatever that is being docked, and again, you can see here template. Now you can see this template APF. APF is atomic property fields. So this particular screening now is going on. Firstly, it is screening with the pharmacophore and then it is docking as well. Now, so it has docked 12 compounds, right? Now again, first day I finished 10%. Then next day also, if I come, I can go here, make another hit list. I can update this hit list to that. And now you can see 49%, you can see 19 compounds are docked. So this way, how versatile it is for generating a pharmacophore and VLS, doing a VLS much more effectively than other tools. So this is something about VLS and normal docking. So we will also calculate the other features for for these compounds. For example, now let us consider these are your own designs. Now, if I wanted to run a tool called Molescreen to know the ADME ADME parameters of that for these compounds. For that, what we can do is go to Molescreen, load ADME, and these are all the different models that are there. And here you can see the data set. So this particular model has been built using 1300. Here it is 2,89,000 compounds. Here it is 1800. So the data set is quite huge and quite big only here for developing these models. And again, there is something called AUC is pretty good. RQ squares, RMSEs, and everything is available here. And if I wanted to calculate for absorption and other parameters, like I'll just check each and every option and then I will select. This is the data table to which I want to calculate these parameters and I will run the Molescreen. As you see, both the jobs are going on, but still it will not take much time to calculate these parameters. So this is the data that is being generated for these compounds. So the CACO2, if it is minus 4.8, it is, I mean, here they are color coded. There are standard reference values available in the manual also. If it is greater than -6, its permeation is not so good. So CACO2 without any coloration, it is 4.8. It is pretty good with this. And to which family it might bind to. So this molecule, this compound, will it bind to CYP? Will it bind to CYP GPCR? Will it bind to ion channel? Will it bind to kinase? So if it is 1 means if it definitely hit that particular class of family proteins. So here it is 0.3. So definitely it is much more having a less probability of binding this molecule binding into the other class of proteins. Log half-life, you can see the values for calculated from monkey, dog, I mean microsomal half-life and plasma half-life. It has finished the job. So let us. So plasma half-life, microsomes half-life, and various other parameters like efflux for apical to basal, basal to apical, and many more. Like you can see MDCK cell lines, it also calculated apical to basal, basal to apical, and also CYP. So this particular molecule, is it hitting the CYP? Yes. For CYP, this molecule is a binder of CYP1A2 and also CYP2C19. So this way we can, before synthesizing them, we can also predict the ADME parameters of those new designs and we can get rid of some compounds which has that particular features, hitting any of those ADME parameters. And on top of that, you can also see whether this molecule is going to be oxidized or not. For that, we can go to mole screen and predict and say, okay. So this will also help us in understanding which particular moieties might undergo oxidation. So in this case, what we can do, we can keep a fluoro over here or a methyl over here to avoid the oxidation. And here, this particular alpha carbon, again, it is telling us that this could undergo oxidation. So this way, metabolic oxidation can also be read. So this is something all about docking. And so this is what it finished the docking. So we got in this particular VLS, you can see this template.apf. And as I was telling, what is the use of two different scoring functions. For example, now I have a physics score. Now I also have a RTCNN score. And then I can plot it like this. And during selection of my molecules, what I can do is, here you can see the physics based scoring function is this particular compound. Actual activity versus physics based, it is 7.7, and its physics RTCNN is 41, but physics based score is close to 50. So my physics based scoring function is misguiding me in this case, right? So this way, this is, as we know, this we have the experimental data this way. If we don't, then we can also select the molecules. Like you can see, this molecule is having a score of close to 44 on score, and also close to 43 with regards to the RTCNN score. So this type of molecules we can select to avoid false positives during the VLS study. So this is all I thought of showing you today.
Thank you so much, sir. It was
Yes.
wonderful because I'm working on this computer drug design since last almost 8 years. So
Yeah.
what I have seen and what I have faced the most difficult challenge that it takes long time. Like after doing pharmacophore screening, then we go for docking, then we calculate
Yes.
this ADME toxicity, drug likeness, all these things. But in your
Yes.
case, with this particular there is a moles of Tysium Pro, you did everything in a just a single few clicks. That's the
Yes.
best
Yes.
part of particular the software, and because
Yes.
it will save you lots of time. And moreover, whatever
Yes.
I have seen, that all those MDCK and capital permeability, all these thermokinetics related
Yes.
parameter already models have inbuilt. So it is wonderful.
Yes.
And RTCM, we
Yes.
already have shown that it has better scoring function than all the existing scoring parameters. So that's truly
Yes.
wonderful. So
Yes.
that's
Yes.
great. So much. Sir,
Yeah.
you also in that brochure also mentioning about the product. So will it be fine to tell few 5 or 10 minutes with product?
Yeah, definitely. I can do that. And one more thing I would like to tell here, there is no
Yes, sir.
ligand preparation in this particular tool.
Yes,
But
yes.
coming
This
to,
is the
yeah, so other softwares, here we only prepare the protein, generate the grid,
yeah.
import the protein and import the ligands and dock. That's it. So the ligands are
Yes,
generated on the fly.
absolutely. For this pharmacophore also, for example, like
Yeah.
Schrodinger, they have this phase and also first have to create the database. It takes long time
Yes.
because having
Yes.
millions of compounds, it's long time for making the database, and this is wonderful.
I know.
Really,
I, I know. And again, if you are working with metals, it will be much more challenging.
yes, sir. Absolutely.
Yeah. Yeah. So, for example, Protac, nowadays everyone knew that this is one of the most happening area, but still there are no drugs in the market. But a lot of research has been going on into this particular domain. And Aravinas is the company which has brought this particular concept of Protac, and Protac usually stands for protolytic cameras. So here, usually what happens, usually a traditional way earlier, if we have anti-cancer drugs, like we used to have, they usually have a lot of toxicity, right? So those
Right.
anti-cancer drugs goes and bind to your target protein and they stops the function of this particular protein. But here in this PROTAC, what is done is your your kinase molecule is connected with a linker. For example, now let us consider this is a BRD4 protein. This is the ligand which is binding to your pocket, and then you extend on a particular point with a particular linker. Maybe it can be aliphatic, or it can be pegylation, or it can be an aliphatic chain with an aliphatic ring systems, multiple linkers. And then on the other side, you connect it to another ligand called CRBM. So what happens biologically? So these proteins, so when we target the proteins with PROTACs, so your protein of interest, this ligand, this is a BRD4 ligand. This is a BRD4 ligand. It binds onto your protein of interest, and this remaining part is in floating in your cytoplasm. And then what it does is this particular molecule recruits the E3 ligase. And again, biologically, what E3 ligases does is they come, they bind to the target protein, and they ubiquitinate this. Ubiquitination process is biologically it ubiquitinates. So once the protein is ubiquitinated, automatically this protein of our interest will go to proteasome and it gets degraded there. So the first therapy only in the anti-cancer, we are stopping the function of the protein. But using PROTACs, we are stopping the function of the protein. At the same time, E3 ligase is coming and ubiquitinating the protein, the cancer protein. That protein is being sent to proteasome for degradation. So this way, the protein itself is getting degraded in the cell, in the body. So that this is the theoretical concept and the biological concept. And again, what are all the different things what we need here? Again, I may not be able to show you the demo of it, but what are all the prerequisites, how to run, what is the setup, I can explain. And I will show you the results file, because this particular job will take maybe 2 to 3 hours for a single ligand to run, because initially it undergoes the protein docking, then followed by small molecule docking. So that way it happens. So here, what are all the prerequisites I would need is, first, let's see two different proteins with bifunctional proteins, means not bifunctional, two proteins with the crystal ligand or your docked conformation. For example, now consider this is my kinase. Now I have a crystal structure of my ligand, or I dock this molecule. That is also good enough, right? And on the other side, we may have to have a E3 ligase, either co-crystal with the small molecule, or you can dock your ligand here also. So these two complexes are mandatory and in the workspace. And in the chemical table, you can see this is my PROTAC of my interest, which I want to dock into this protein. So these three things are mandatory, and the setup file looks pretty easy and which a very straightforward methodology. We'll go to docking, protein docking, PROTAC model builder. Here it is target protein. What is my target? What is my ligase? Here you can see my ligase is 3MXF and my target is the 4C1I is my ligase and target is my 3MXF. So this is my protein of my interest. This is the ligase, and we will give the PROTAC table. This is the name of the ligand, and this is the index, means one ligand, one. For example, if you have 10 molecules here in this table, so you can give the number. For example, if you wanted to dock the fifth molecule, you can change the index and you can keep the effort one here. Effort one is good enough in this case. And again, number of parallel runs. By default, it will take 5 jobs parallelly. This particular docking, as we know, it is very computationally more extensive. That's why it takes more time to run this job. So it is by default 5 and we can hit OK. So this will take up the job and it will run the job. So these are all the prerequisites and the setup what we would need to run a PROTAC modeling. So if you have any queries here, doctor, we can look into them. Else I will show you the results file as well. How to
So the
analyze them.
linker will be also protein, or the linker will be some small molecule, or what?
Linker should be on your small molecule. For example, you can see in the graphical window,
Yes.
I'll off all the ones. So now this is my BRD4 domain protein with particular ligand co-crystallized, right? Now
Sure.
I have E3 ligase co-crystallized with another protein,
Correct.
right? Now I have to connect these two. Here you can see the linker. This linker is coming from this thiol ring on my ligand from here.
Right.
Here. Because it is completely on the solvent exposable area. If I choose any other side linker, it might bump out. So we have to select the exit point in such a way that it is completely in the solvent exposure. Here we have built a aliphatic chain, and again this chain is being connected onto your molecule of the other side. Here again, this also you can see they are targeting the aromatic ring here.
Right.
Then this is the molecule which we are expecting. And again, it is not 100% mandatory. Like this particular point only, you might find a right linker. They keep changing the positions of this linker also on this aromatic ring. And also they keep changing the linker size also. So which linker site would be a best one? So that you will know after biological test. Here also you can understand with regards to docking and also with regards to the degradation. If the degradation is much better in biologically, we can consider that particular linker with that particular molecule is much better. So this way it usually happens. So I'll just try to, we'll try to analyze the uh, results of this. Now, yeah. So this is the complex which it made now, right? Now,
Wow.
it generated a stack of 1100 conformations. Because protein usually, like here it is a protein docking. We cannot just say like only one conformation it could bind to the target protein. It's not like that. So it might run like this. So it would generate multiple conformations of the other side of the protein. One side of the protein is static here. So this way it generated multiple conformations, and we may have to select the right conformation based on some energetics and some scoring functions. So this
Sir, I have a
way,
question.
yeah.
The blue one is the target protein, that is the kinase, and the another one is the protect protein, right?
The green one is our target protein of interest. This
Right.
is our E3 ligase. Gray one.
Great. Wonderful.
Yeah. So now here you can see there are not many parameters, not many things. Here one is the energy, the total energy of this particular system. And this is the comment. So if we select this particular option, that particular pose is loaded into your graphical view. You can see if I change it to something else, it changes from this table.
Right.
And this is the electrostatic energy. This is the surface energy, van der Waals energy, electrostatic energy, and this is the energy of the side chains. Now, how do I select? Usually what happens, we cannot take just one particular conformation, consider this one conformation to go forward. Usually it is suggested to take 10 conformations, top 10. We can plot them with regards to energy versus energy of the side chain or anything else, and then we may have to select them. This way, we may have to do a lot of due diligence while selecting these poses. And that particular confirmations, we may have to run MD and see whether this particular complex is molecule and this complex is staying together. So that way we may have to validate the process.
Great, sir. That's
It's,
truly wonderful.
yeah, it's a really very, very straightforward approach. And again, with my experience, what I have seen, I've seen one particular PROTAC structure where it has multiple chains, multiple protein of interest chains, multiple E3 ligases. Means chain A, chain C. Let's consider chain A and chain E, chain B and chain like that. Multiple complexes. When I try to overlay uh, two complexes, I saw a huge deviation in the crystal structure only.
Okay.
Yeah. So that's why it is very, very hard to predict the correct right confirmation. Yes, but unlike other tools, other softwares don't have this particular module. MoleSoft has, and it comes with your ICM Pro. So any questions from the student side or any questions
Students,
from other faculties?
students and faculty members, if you have any question, please raise your hand so that I will make you the presenter so that you can ask the question, because in by default I have kept all the attendees as muted, and also your video is not visible. So if you have any question, please raise your hand. I will make you the presenter so that we can interact directly to Professor Shamshed Singh Sardar.
So there
Okay,
are no
so what if
questions in the chat as
there
well.
are no questions? Yeah, there are no questions. So
Yeah.
thank you so much, sir, for this wonderful today's lecture followed by the software demonstration. And this is truly incredible. Based on my experience, I can say this is the state-of-the-art technology that you are introducing in India. So thank you
Yes.
so much, sir. We'll be giving you one certificate after taking this signature of our honorable director, sir. We'll be sending you
Right.
the certificate through Dr. Shitej. So
Yes.
thank you, sir. It's a great
Thank
time to
you.
talk with you.
And
Yeah,
yeah, Dr.
yeah,
Abhijit, if you would like to evaluate this particular tool, you can just get in touch with Kshitij and he can help you in evaluating the software. I
sure,
mean,
sir.
there is some formality like you may have to, I mean, he can guide you in that. So you can evaluate this tool and later you can think of other process, other things.
Right, sir.
Yeah. Thank you
Sure.
all.
Okay.
Thank you, everyone.
Our session.
Thank you, sir. Thank you, everyone. So have a good day, sir. See
Thank
you
you.
soon.
Thank
Thank you,
you.
everyone. Thank you, Rupa ma'am, and thank you, Abhijit sir.
Thank you, Shitoj, sir.
Thank you. Thank you.
We're ending our this today's webinar. Yes.